Thank you, Ewan, for your kind introduction. Hi, everyone. My name is Hsiao-Chi Liao, and I'm a PhD student at the University of Melbourne. Today is my pleasure to share our research with you today, and I'll be talking about a toolbox that we developed to analyze the data from massively parallel cytometry experiment. So I will start by introducing single-cell proteomics, and then to talk about what is massively parallel cytometry experiment. Then I'll show you some evidence of the background noise and well effect in this kind of data. Then I will talk about the workflow for analyzing this kind of data set, including our method and existing pipelines. Then I'll show you some results and the take-home messages. So in our study, we focus on single-cell surface proteins. So those are the proteins on the surface of a cell. And we know that the heterogeneity of the cell surface proteins helps to identify the cell populations. And there are more than 400 CD markers can be used to identify different kind of cell types. But there are some limitations in terms of the number of markers that can measure in a single experiment. Like, for example, the conventional flow cytometry, the fluorescence-based method, the number of markers can be measured in an experiment is around 17. And for cytoph, it's up to 40. So to get more protein markers measured, there's a new assay. It's called MPC. So this allows us to measure more than 200 proteins in one experiment. And there's one commercial brand. The assay is called Legend Screen Assay, and that was from BioLegend. So the advantage of using that is that it's high throughput. So it basically allows us to have simultaneous analysis of multiple samples, and it's cost effective because it can lower down the experimental cost. And the inflow protocol here is the pioneer of producing this kind of data and to analyze MPC data. So the experimental pipeline starts by staining all the cells, all the single cells with backbone markers. And the backbone markers, they are basically the markers, they are really informative, and we can use it for broad for broad cell type recognition. And the second step is that we have this plate-based assay, and in each well, there's one particular protein marker. It's called infinity marker, and it's kind of exploratory marker. So this is an illustration of the cell from different wells. Apart from the backbone markers they can measure, and we have the Okay, so we have different kinds of infinity markers can be measured depending on which well the cell came from. And then we acquire flow cytometry for each well. And the computational pipeline, how do we analyze the data from this assay? So as we can see, the trick is that we get to measure a lot of protein markers. But the problem is that we only have the cells came from that particular well with the measurement of the particular type of infinity marker. And we have the measurement for backbone markers for every single cell. So cells are on the rows. So the computational pipeline, the key here is to impute the unmeasured infinity markers for the cells in all the other wells by using machine learning methods. So by taking the information of backbone markers, we are able to impute the unmeasured infinity marker for the other cells. So ultimately we will get this kind of the completed data matrix for the downstream analysis. Yeah, and like many other omics data, the unwanted variation exists in this data set. So here I'm going to show some evidence. Firstly, here is the distribution of a particular marker. And we can see that some cells, they have negative values and some cells, they have positive values. So one key processing is that we need to do data transformation for later easier modeling. And basically, the background correction is needed because every single observed intensity is a combination of signal and noise. So this is an example that we measure the noise in isotope control well. And then we do need batch adjustment because the systematic effect exists in the raw data. So this is an example that we have the summary statistic for each well from different plates. So we have 3 plates, and you can see really strong well effect within each plate and plate effects. So we need to deal with that. And even if we perform background correction, the systematic effect still exists. So this is the evidence that we see the well effect, strong well effect in the background corrected data. So in terms of analysis pipeline, our method is called MAPFX, and there are 2 other alternative pipelines. So I'll start by introducing the current method and talk about their problems. So firstly, the current method is called, is from the info protocol that I showed earlier. And you can see that at the first step, this method and then other alternative method, they all start with data transformation. It's called logical transformation, and it has its own problem. I'll explain that later. And the second step is to do batch correction. The info protocol, it uses Z transformation to achieve that goal. And other alternative pipelines by applying combat, a more sophisticated model to remove the well effect. Then continue with imputation by machine learning methods. Then finally is to perform background correction. So this is the current pipeline, and we are not happy with that because firstly, when we look at logical transformation, you can see that for the large values, actually it's like log-like transformation, but for the small values around 0, it's linear-like transformation, and it's inconsistency for a single feature, and we don't like that. And the batch correction, the problem of using that transformation is that that will remove the feature-specific variation. And combat is that the problem is the background correction does not include it in that pipeline. And finally, the problem of doing background correction after imputation is that because imputation will introduce uncertainty. So if we do background correction after imputation, then it's not the ideal way to do that. So instead, our method, we improve the way to perform background correction and removal of unwanted variation. So MAPFX starts with background correction by applying normal exponential convolution model. And the second step is that we perform data transformation by using the consistent transformation, that is the log transformation. And then we perform batch correction by using the log normal model. And finally, after we cleaned up the data set, we continue with imputation. So if anyone is interested in our technical detail, you're welcome to check out our preprint for technical details. Okay, so the results. One thing that we want to assess is that after correction, data correction, what is the preservation of biology and the leftover unwanted variation in the data set. So here I'm showing the silhouette score. A good silhouette score for biology here in the example here is the cell type. So we want the value to be higher. And our method in pink did outperform others. And in terms of leftover batch, 3 methods that overlapped at the bottom. That means we remove the batch effect equally well, but we preserve more biology than others. And in terms of using our corrected data to perform imputation, we do have better imputation results. As you can see here, I'm showing the R square, and you can see that our method have a higher median, and the distribution is better towards 1 than other 2 methods. And if we check the infinity markers 1 by 1, the imputation result, then you can see that our method is actually having a higher proportion of infinity markers have better imputation results. More than 70% of the infinity marker, we have better accuracy in terms of imputation. And also, this is very exciting because we see that the CD4 T cell, using our method, we have the potential to find the subpopulation of naive CD4 T cell. So this is evidence that we see the potential of finding the subpopulation. And the key marker here is Ly6C. And also, I found some paper that suggest that Ly6C might be the potential marker for finding subtype of naive T cell. And another example is B cells. Here, using our method, we have the ability to separate T2 B cell apart from T3. So our tool is publicly available on Bioconductor, so you are welcome to download it. And if you don't have this data from this assay, if you have data from conventional flow cytometry assay, you can use that as well. And we have the VNet provided. You can follow that to do your data analysis. And for more technical details, you are welcome to check our preprint on bioRxiv. So the take-home message, why do we want to use MPC experiment? Is because it's the characteristic of high throughput and low cost. And why using our tool is because it can effectively remove the unwanted variation, and it's free. So, and I checked the Google Scholar 2 days ago, and I found there are more and more data popping out at this moment. So that means there's a need to have a good analysis tool for analyzing MPC data. With that, that's the end of my talk. Thank you for listening, and I also thank my supervisors. This is a joint work with my supervisors, Agus, Terry, and Davis, and also my lab mates, and my collaborators, and scholarship from University of Melbourne and Taiwanese Ministry of Education, and travel fundings from ISCB and the University of Melbourne. Thank you. Okay.